Okay, so I'm going to go through the questions that I asked you to complete. So question 1, it says the amplitude, that's the height, and the duration, that's the length, of the action potentials in a single neuron are always the same. However, the amplitude and duration of action potentials can vary between different types of neuron. So this is worth 6 marks. And they have given us 2 different types of neuron. So the top one is a Purkinje neuron, so that's going to be from the heart, and the bottom one is a dopamine neuron, which is going to be from the brain. Both of them have time running along the x-axis, and they have membrane potential in millivolts on the y-axis. So the question says, describe the differences between the 2 action potentials and suggest explanations for the differences you have described. Well, we know that action potentials are really caused by sodium ions and potassium ions moving in and out across the cell membrane of the neuron, and using sodium potassium pumps, or by using voltage gated channels. And so any differences between them are going to be caused because of those channels and because of the pumps. So let's first of all write down the differences between them. So the first one that we could look at is the let's look on the y-axis first actually and look at the difference between them. So this one here, okay, the dopamine has actually got if we look here, okay, you can see that it's got a resting potential here at the beginning, which is much lower, or no, actually higher, sorry, than the one on the Purkinje neuron. So that's going to be my number 1. Okay, so I'm going to write here. So difference number 1 is that the dopamine neuron has a higher resting potential than the Purkinje neuron. I'm just going to check how to spell that. Okay. So difference number 2. Okay, let's have a look at the next phase of the graph. So the second phase of the graph, we have here, we've got the action potential here reaches 10 millivolts, and in the Purkinje it reaches, probably, if I had a ruler I would draw across there, probably 20 millivolts. Okay, so the dopamine neuron has a action potential of 10 millivolts. The Purkinje neuron has an action, can't spell, of 20 millivolts. And I could also look at how far they've risen. So the action potential changes by, so how much is the dopamine one gone up? So it's gone from minus 50 to 10, so that's minus 50, so 60 millivolts in the dopamine neuron, and it's gone from minus 75 to 20, so that means it's gone up by 95 millivolts in the Purkinje neuron. Okay, so can you see that for each one of these, as I write one answer, I write the opposite answer for the other one, and at A level you have to have written both sides to get 1 mark. Okay, so I'm going to add in some other ones now. I can see that there are there's no clear refractory period with the dopamine neuron. Okay, and there is in the Purkinje neuron. And what I mean by that is, actually, if I look here, it doesn't actually reset itself ready to go back, ready for another action potential. Okay, and also it's much slower as well. So repolarization is much slower. The action potential, if I go back and look here, the action potential is longer. So this one is in the dopamine neuron compared to the Purkinje. Okay, and I'm also going to put here that the action potential is longer in the dopamine neuron compared to the Purkinje. So I've got loads and loads of different answers there showing that I can recognise the difference between each one of those action potentials. Okay, so students lose marks there really because they only pick one half of it and don't give the other side. So now I need my explanations. So as I said before, really when we're looking at explanations here, we're looking at those sodium-potassium pumps, and we're looking at the channels and the movements of those sodium ions and those potassium ions. So what could be different about them? Okay, well, there could be a different number of sodium potassium pumps. There could be a different number of sodium channels, or sodium ion channels, don't forget that bit, or potassium ion channels. If we've got on this, if we go back up here and have a look at the picture, here we know this one was much steeper. Okay, so we had a more rapid flow. Actually, we didn't mention that here, that this is steeper than on the dopamine one. We could actually say, therefore, that if the ions are moving more quickly, there might be a steeper gradient. So it might be a steeper sodium ion gradient in the Purkinje neuron, okay, as graph is steeper. Okay, so I've got plenty of things here that I'm able to write to gain those extra marks. I could say also that I've got different types of voltage gated sodium and potassium ion channels, but any combination of those would give me the marks. Okay, right, let's go on to question number 2. So question 2 shows me a table, and I have to write down the answers to these statements. So I have to write an S if it's sensory neurons only, M if it's motor neurons only, and a B if it's both sensory and motor neurons. So transmit nerve impulses from the CNS. So the CNS, I know, is the central nervous system. So if I remember my SRM, school's really mad, then I know that from the central nervous system, that's the relay neuron, it's going to be going on to the motor neuron. So the answer to this one is going to be M. The next one, connect to other neurons via synapses. Well, all neurons do that, so it's going to be both. And then connect to effectors. Well, that's going to be a muscle, and I know that my motor neurons carry information to muscles, so the answer is going to be an M. Okay, question 3. The skin is important in enabling the body to detect changes in the environment. This allows the body to respond to these changes. Various types of sensory receptor are located on the skin. So figure 1, they're showing me the transverse section through a pterygium corpuscle. So there it is. Okay, we've seen that before, we know what that means. Figure 2.2 is a longitudinal section through a pterygium corpuscle. The tip of the sensory neuron is not covered by the myelin sheath. Okay, again, we know what that is. And it says to us the pterygium corpuscle is a type of biological transducer, As a pressure stimulus is exerted on the corpuscle, the lamellae are compressed and exert pressure on the tip of the sensory neuron. The plasma cell surface membrane on the tip becomes deformed and more permeable to sodium ions. Okay, and we know that that then triggers that action potential. This region of the neuron becomes depolarized, reaching the threshold potential, and an action potential is generated. Why is the pacinian corpuscle described as a transducer? Well, a transducer is something that converts from one form of energy to another, and in this instance it converts mechanical energy into electrical energy, which is our impulse. So defamation of the plasma membrane at the tip of the neuron causes the membrane to become more permeable to sodium ions. Suggest why? Basically because the channels will now start to open. So we are going to have sodium ion channels open. I could say that also because the membrane is being squashed, perhaps I'm going to get some spaces or some gaps in my phospholipid bilayer, and that might let some in, but we know that the channels will open when they become deformed. Okay, part 3, the generation of an action potential follows the all or nothing law. What does that mean? What does that mean? It means that we have to reach a threshold value for depolarization to occur. And if we don't reach the threshold, no action potential is generated. Okay, describe how information about the strength and intensity of a stimulus is communicated to the brain. Well, we know that an action potential is going to be the same for every single neuron. It's the same voltage or millivoltage. So actually what is going to be, how is the brain going to tell those two things apart? How is it going to know that something is more intense than another? So the only idea really is how often the brain receives the action potential. So it's going to be the frequency of the action potentials. And we know that the more frequent would mean a stronger or more intense stimulus. Okay, part B. When clothes are first put on the body, a constant gentle pressure is applied to the pressure receptors in the skin. After a short time, action potentials are no longer generated unless there is a change in pressure as the clothes move over the surface of the skin. Suggest an explanation for the fact that action potentials are not generated constantly while wearing clothes. So what's happening there is that the very gentle pressure is keeping those sodium ion channels open. Okay, so the sodium, let's write that down, sodium ion channels remain open, okay, which means that we then aren't constantly able to trigger that full action potential, because we need that influx to cause that action potential, and if they're just constantly dribbling in, then we're not going to get that full action potential. So sodium ions are therefore in the wrong place because they're kind of constantly trickling into the membrane, so we can't then have that rush of them coming in. Sodium ions are therefore in the wrong place to trigger